Okay, so the rationale for this. So these are data that you've probably seen before. I mean, that's basically what you plotted where you have all your cells and then with different conditions, just like I think conditioning for the animal, CS plus, CS minus. And you can see like there's variability in the cells. Potentially there's some clusters that activate a lot and then some that are moderate and then some that suppress. But then if you average across all the cells, it's that structure, yeah, the variability kind of goes away. The question is, can we group these cells in a relatively unbiased way compared to like, let's say doing t-tests of this window versus that window? But basically, can you take into account the activity across CS plus and CS minus and see if there's any groups of activity or groups of cells that have different activity? That's kind of like this as well. This is just a very basic illustration of k-means clustering, not really what it's doing, or not really like the underlying mechanism, but rather what's doing where you have your data points, like each of these data points could be cells, and then the cells are being described by certain um, we'll call features or attributes. Um, and these attributes can be like activity. And at first it gets maybe you've thought about this enough that it's not it's more demystified, but when I first thought about this and I think when a lot of people first think about it, it's kind of not clear or it's hard to think about because and I'll try to show a little bit more about how this is working, but like these axes are basically like time point one's activity and time point two's activity. Um, yeah, again, I'll kind of explain what that all means. But yeah, again, the idea is to group the cells based on their activity dynamics. So this is kind of a broad overview of the steps. You probably already know to a certain degree where um, first off, we want to prepare the data to compute trial average activity. It's partially the reason why we do the trial averaging is because if we didn't, then we'd have the data would be in the format of cells, time, and trials. But these algorithms they like need to have like two-dimensional data. So we're kind of simplifying doing average across trials. I mean, if you're interested in the evolution of activity across trials, that is something there's other algorithms that you can use to cluster based off of that, like more multi-dimensional data. And then you might be also wondering, well, how do you do that if you have multiple conditions? Because that's also another dimension. And here, this is kind of a uh, cheat, I guess, a roundabout way of doing this, in that, and I'll show you like more visually how we do this, but it's to basically take our trial average activity um, for your different conditions, and then just kind of stitching them together. Um, So then once you have that data, those data prepared, we want to pump them through the actual machine learning kind of algorithms but before we get to the actual clustering, we this is kind of an optional step in the grand scheme of things, but it I think it helps. Is to perform PCA dimensionality reduction. Are you familiar with this method? Okay. Um, and I'll explain a little bit more. But really, the main reason to do this is just to make the data more amenable to clustering. Because I think I mentioned to you in that these clustering algorithms they prefer when there's fewer features describing the data. So fewer time points that describe the data. And then there's things called hyperparameters, which are um, I guess parameters, for lack of better words, um, that I should have prepared this stuff. What are hyperparameters? Let's look this up briefly. I have this. I have this. Oh yeah, okay. So it's these are just like variables, values that not so much, and they're not the variables that get that are learned during the like the training and the running of the model, but rather they're the input variables that the user can put into to influence the learning and the training. Um, but basically, we're going to want to supply the whole pipeline with a set of like multiple parameters, hyperparameters for it to test and to get the optimal values. And then once it gets the optimal values, it'll do the final clustering, and then we get the visualization. Okay, so preparing the data. So this is basically like imagine like the actual data that we have um, that we prepare in the code, where it's our cells on the y-axis, and then this is like for CS plus, this is like time points on the x-axis. So essentially, you get like the activity traces, right? This is average across trials. And then like at some point here, maybe like sample six is where time zero is when the event comes on. So you also have something similar to like for CS minus. Note like the dimensions are similar. And most importantly, it's the Y dimension has to be the same. Because if you're trying to cluster cells, they need to be like the data should be coming from the same sources. So the idea, remember how we were talking about stitching the trial visualization together? We're basically just gonna do that. And now before, remember there's 51 time points. Now it's 102 because we kind of combine them together. That's exactly what the code's doing. Um, so now with PCA, this is my kind of attempt to kind of explain this. The first way for me to introduce this is a little bit more intuitive. Let's just assume that these data points are still cells, but our initial X and Y axes are like gene one expression, gene two expression. So like this is like if you pick a cell out here, then it's like high in both expression patterns, right? That's relatively easy to to understand. Um, but for us, it's not gene expression, but rather it's going to be time point one and time point two. In in actuality, it's because this is a very simplified like representation, right? Because we have 50 to 102 time points. So the space that the data occupy are actually like you can't even visualize it. This is just like our two-dimensional, very simplified representation. But when we do our clustering, kind of depending on how many principal components, and I'll explain what that means in a bit, that we end up with, it's going to be more complex spaces. Um, and then hopefully this representation right now will demystify both how the cluster, like what the data, what the clustering algorithm is applying to the data, how it's interacting with the data, as well as what PC is doing. And hopefully some of this is a review for you. So let's see, okay, so this is kind of setting the stage. So again, we're thinking about the time points. These are individual cells. So this, if we choose a cell that's out here, it means that here, it's active at both time points. So like basically all the cells out here are gonna be in a sense similar because they're all activating at both time points. And then you can also imagine like if you had a third dimension and it also was grouped up in there, then that means like the cells around there also have similar activity dynamics. Um, so that's essentially the kind of the underlying kind of assumption here when we're doing the clustering in this space. It's that when data points are, because our axes are the time points, um, and then where they are in space kind of dictate how active they are at the different time points. We can then the cells that are kind of closest together mean that they have more similar activity patterns. So kind of the opposite here. These are cells that are kind of lower in activity, these time points. And this is just some, I mean, this data point doesn't probably exist, but um, this one has a different kind of dynamics. One of the key things that I want to highlight in terms of the PCA part is that you see that a lot of data points are kind of lying along this kind of diagonal. The idea of PCA, so this is all before PCA, but with PCA, it's a statistical test to find like similar patterns in the data and to find a new set of axes to plot the data into. So basically, we're not super interested in these original axes anymore. We're going to use this new set of axes that PCA has kind of discovered. And the reason why we want to do this is because so PCA is trying to find new axes that better explain, that capture the variability of the data. Because like here, our original axes, to be able to like figure out to define a cell, to define a point, you'd have to know both time points, like activity, right? But here in this one, because most of the cells are along this axis, you could just be like along this axis, it's the cell is like out here. And then just by using a single axis, you know that it's less of the contribution is from this axis and it's more so along this. It's kind of nebulous, but hopefully that makes a little bit more sense. And then in that case, because this axis here is not so important, we can just say we're not interested in that axis. We're just going to throw it away. Um, so that's kind of how we're able to decrease the number of features that explain the data. Because here, whereas we needed two axes, two features to identify a cell, now we only need one. And okay, we kind of already talked about this. Because now our new axis one, PC1, basically we go to the right, it means that the cells are active in both time point 1 and 2. But if we go to the left, it's lower in time point 1 and 2. And then if we go on this other axis, it's like these opposite dynamics, but because there's not that many cells along that axis, it's not that important. Okay, so this is, I mean, you've seen these plots already, so this is kind of the result of the PCA. You might be wondering how the algorithm determines how many PCs to keep, how many to like throw away, right? Keep. Basically, it's plotting for each of the principal components, kind of ordered in order of like how much each principal component, each new axis explains the data. And it's kind of a typical procedure to either just take the number of PCs that like result in um, or add up to where it bends in this plot. Or another way to do it is to keep the number of PCs that explain 90% of the data. So like the other 10% is probably maybe due to noise or some uh, random like dynamics. And then these are just kind of uh, this is not super important for you to, I guess, know like when you're running things, but it's how for a given principal component, how how much the original axes contributed to that. So also kind of what each principal component is like the overall um, activity pattern. Uh, Patterns are encompassed using that. Does this kind of make this? Yeah, it makes sense. Okay. All right. So enough of that. Enough of that boring-ish stuff. Well, I find it kind of interesting, but so we want to. So this is our reduced set, right? Because here it said that we want three PCs. Well, I guess we have four here, but now instead of having 102 uh, columns to explain the data, because remember this is cells, and then we want a cluster based on like how what these numbers, the patterns are. And now we only have four, so it's like way easier or way more amenable to go into k-means clustering or spectral clustering. So now, what is spectral clustering and what's k-means clustering? I think here I'm gonna well, let me just think of start with. Maybe I start this first just to kind of set the stage. K-means clustering. Like this is I just ripped off from Scikit-learn's website where it's kind of showing you for different like data kind of blobs how k-means clustering and this spectral clustering works. This is probably the most like similar data sets that we'll be working with, right? Because like none of like our neural data really look like this. Um, so like kind of Gaussian-ish blobs. Um, K-means clustering is trying to group data points based on how far they are in like actual space. Whereas spectral clustering, which I'll explain what it's doing, is based on like kind of more graph theory. Also, in a sense, um like looking at uh closeness of proximity of data points, but more so looking at like how many neighbors are like uh close to a certain data point. I guess go back here. So k-means clustering, do you know how it works? Okay, so this would be helpful. It's actually really intuitive and it's a very elegant algorithm. So these are steps. So like these are your data points in space, right? Time point one, time point two again that we with. Or actually PC1, PC2 now. So step one is basically initializing starting points. And these could be anywhere. Like um it's just kind of random where it's initially when it's set. There's a parameter to like lock these into specific points, but in this case, it's going to be like this. And then what it's going to do in step two is to group the data points based on how close they are in space. So like all these blue ones, well, all these ones right here are closest to the blue, so they've been labeled as blue. And then these ones are closest to the green, so they're labeled green. And then now it's going to recompute the centroid. So we're calling these like uh the center of mass like point is called a centroid. So that's basically just taking the mean location. So now instead of here, it's just shifted to here. But because it's shifted, because these centroid positions are shifted, now the relationship of data points on how far they are to each centroid has changed. So then they do kind of reclassification, right? Because now all of these are close to this one, all these are close to this one. And then they basically just uh repeat those steps until there's like very little movement of the centroids. And then once it's stabilized, that's something things don't make sense. And the let's see, how are we doing on time? I don't want to spend too okay, getting close. Um, the number of centroids, because you can imagine like there could be more than two clusters. One of the hyperparameters, which I'll highlight in a bit, is the number of clusters. And as we kind of modified that in the code. Where I told you to change it to 10 or 20 like that. So that number should be how many clusters you expect to be in the data, which you can kind of get a sense for just by looking at the raw data, right? Okay. So I think I'm just going to kind of broadly explain what spectral clustering is doing. It is a little bit more complex, but there are other resources there to inform you. I'm going to more explain the kind of parts that are relevant to the hyperparameter adjustment. So again, it's a graph theory based sort of approach where like imagine the data points in our normal space, the PCA space. What the first step is doing is graph creation. So looking at how many, like for a given cell, how many data points are kind of closest to it. I guess for this one, it's you can say like these ones, but the hyperparameter here that's important is n nearest neighbors, which is like if you ask for 2, then it'd be like these two data points are the closest. So for 3, there's like these kind of connecting lines. And then because of like you have all these connecting lines that indicate if they're connected or not, you can make a matrix that indicates whether they are connected or not. So here you can see that for cells for number 3, it's connected to 4, so there's a 1 there. So this is like it's called the adjacency matrix. You don't really need to know like the name of it and such. But then it takes that data and then it performs this eigen decomposition, or it generates a Laplacian matrix and then or performs eigen decomposition. So let's see, I have some notes here. Just okay. So the Laplacian is just another way to represent the graph that gives a little bit more information about the connectivity. And then the eigen decomposition of the Laplacian matrix, a lot of fancy words. It basically, if you do it, it highlights the kind of differences between tightly and weakly connected nodes. And that's going to facilitate like the clustering. So then once we have that information, then it's a little bit simpler. It just runs that kind of simplified data relationship in through kmeans clustering. And then we already talked about kmeans clustering. So with this overall structure, we've actually found that it does a better job than k-means, or at least, I mean, based on this kind of like representation, and then a previous postdoc, BJ kind of consolidated onto this method. Okay, so hyperparameters, we already kind of talked about this. Oh, okay, so I had the demonstration. Should talk about this earlier. But it's an n clusters, which is going to be that's going to be for both k-means and spectral, and then n neighbors. Again, we kind of talked about that again. So I talked about that to a degree. This is pretty straightforward to parameterize. And then this one's a little bit more trickier, but I have some like guidelines in the code. Final thing in this presentation is a silhouette score. So when you run the thing, right, there's like a section where it's like you might have noticed it spits out silhouette score. So silhouette score is a kind of a secondary metric to evaluate how potentially good the clustering is performing. It is kind of roundabout in that it's using distances between data points to evaluate, but even but the algorithms themselves also use distances, right? So I think you have to take it with a grain of salt. But it is what we use to evaluate what's the best hyperparameters. So essentially what it is, but if I can demystify this a little bit, basically for a given data point, you're going to be calculating the distance, average distance between that data point and the data points within that cluster. And then also for the other nearest cluster, it's calculating distance from that data point in our cluster 1 to those other data points in the cluster 2. So you can imagine if clusters are tight and they're further away from other clusters, this number will be small and this number will be big. And so that's basically what's happening here. It's just kind of like combining those two data to those values and then normalizing. Essentially, this is the most important thing, is that if it's close to -1, then it's kind of poorly clustered, and then 1 is it's good clustering. Okay, and then the final thing, we're getting close to time. I'll just try to fit stuff into 2 minutes here. It's fine. I guess we don't necessarily need to talk too much about the actual plots. I can just do that offline. You already know. But we know that these are the hyperparameters. So again, this should just be the maximum clusters expected. So what the code will do, it'll cycle through 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 clusters. It'll test for all those combinations on which one's best. And then it'll also, in combination, do the n nearest neighbors as well. That's only for spectral clustering. So as you can imagine, the more cells you have, the more like possibility that you'll have like other cells that are kind of close together. So this number should kind of, in a sense, scale with the number of cells you have. Again, so
code automatically does kmeans or spectral?
it does both.
Where
So
do you change?
well, no, like you don't change anything.
It
It'll run both of them, and then if it determines that k-means clustering with whatever combination of hyperparameters, or actually k-means clustering is just max clustering. So whichever number of max clusters is optimal. If k-means clustering beats out any of the other combinations for spectral clustering, then I'll go with that one. Let's see, what did it do? Want to do for this one? So here. It says 3 clusters method spectral. So spectral is better. I think most of the time it's gonna win out. But currently there's not a way to just like lock it into spectral or k-means, but that should be a pretty easy thing to. Okay, so that's mainly it. So I'm gonna go through how to run this PCA and clustering code that I have here. This code here, you can get an explanation of the user-defined variables to adjust for. Here, go through each of these parameters. Here, this is just importing the packages. You just run this, and then we have the user-defined variables in this block of code here. So typical things are setting the file paths. You can replace this with an explicit path here. And again, make sure you have the R here to render this as a raw string. we have our primary important parameters here. Here, this is for the trial snipping. So if you're interested in different time periods relative to the onset of different events, you can specify that here. So this currently is saying I want -2 seconds before event onset and then 8 seconds after event onset to be considered for clustering, and then also the baselining period. This is purely for sorting cell activity. And then here we can set the method for determining the number of PCs. We talked about the screed plot method as well as the 90% variance. And then I've already run this, but just to kind of take a look at things, this plot here is actually before any of the clustering or the PCAs occurring. This is just showing you um, the activity of cells before clustering for these different conditions, as well as the mean across the cells, and basically illustrating that there could be different clusters here. Here, just telling us this number of PCs, and then we have our scree plot to determine the number of PCs as well as the principal components and their kind of weightings of the original axes. And then now we're doing the actual clustering. You'll see a bunch of these rows showing the different parameters that it's testing, right? So 4 and 1, 4 and 1, number of clusters and number of nearest neighbors, as well as the resulting silhouette score. So it's going to go through all those different combinations along with the k-means clustering and spectral clustering. And then here, this is what the algorithm converges on in terms of the hyperparameters, as well as the resulting silhouette score. So this should be the highest silhouette score amongst all of the parameters combinations tested. And then it re-performs clustering using those optimal parameters, and then we see our clusters here. So this is doing a moderate job. Ideally, we would have more cells so that the algorithm could perform more often. And then here we have the trial or the cell average responses across conditions. We can also observe how the data are clustered in t-SNE space. t-SNE space, t-SNE is basically taking our PCA, our data points in our PCA, and just kind of rendering them in a space that's better preserves um, the closeness of data points for visualization purposes. And you can see how those data clouds are kind of oriented in space.